Coming to that of the second question, that is balanced equation of nitrogen fixation. Okay, so coming to that of the nitrogen fixation, first let me put down the equation of nitrogen fixation. Nitrogen plus 8 protons plus 8 electrons plus 16 ATP molecules in presence of an enzyme dinitrogenase results in the formation of ammonia plus H2 plus 16 ADP plus 16 PI. So this is the equation of nitrogen fixation. Okay, so let me explain this nitrogen fixation first of all. Coming to that of the nitrogen fixation, and in exactly to speak, this is the balanced equation of biological nitrogen fixation. Biological nitrogen fixation. Okay, so here nitrogen which is present in the atmosphere is usually present in the form of dinitrogen, or in the gaseous state we can say. Okay, so the atmospheric or the nitrogen which is present in the atmosphere is in the form of dinitrogen. Okay, so this nitrogen, if at all, it has to, or it is fixed with the help of the microorganisms, then you call it as, or then you biological nitrogen fixation. Okay, so the reduction of or the conversion of atmospheric dinitrogen into ammonia, okay, N2 into ammonia, that is NH3. So the conversion of or the reduction of atmospheric nitrogen into ammonia by microorganisms, by the microorganisms or prokaryotes, you can also call them, or prokaryotes, is called as biological nitrogen fixation. So simply biological is nothing but which are going to involve living. So living here are nothing but the microorganisms which are involved in the fixation of the nitrogen. What does it do? This nitrogen is converted into ammonia. So that's what we can define. And what are this? And the microorganisms which fix the atmospheric nitrogen into ammonia are called as, they are called as diazotrophs. They are called as diazotrophs. Okay, and this process can also be called as diazotropy. This process can also be called as diazotrophy. So that's what this is. These are the microorganisms are called as diazotrophs, and the process can also be called as diazotropy. Okay, so what happens over here? Here the atmospheric dinitrogen, it is converted into ammonia by taking the power, okay, which is used over here is 8 protons, 8 electrons and 16 ATP molecules in presence of an enzyme dinitrogenase, which converts nitrogen into 2 molecules of ammonia plus H2 plus 16. As ATP is utilized over here, automatically the byproduct which will be formed is ADP, adenosine diphosphate, and PI. PI denotes the inorganic phosphate. So these are the byproducts we can go with. The main end product is ammonia we can deal with. Okay, now. So now where from these assimilate or these electrons, protons, electrons, ATP. So these power are going to come from that of a different mechanism again, wherein they come from that of pyruvic acid. Okay, so here pyruvic acid serves as an source for the source of protons and electrons, and ATP will be coming from a different source. Okay, so pyruvic acid, where does this pyruvic acid, and you are well familiar of the process that is called as glycolysis. You have studied under respiration. Okay, so the end product of glycolysis is nothing but pyruvic acid. So the pyruvic acid undergoes phosphoroclastic cleavage. It is called as, it is a division, phosphoroclastic cleavage, breakdown it is. Okay, so during this reaction, the end product of glycolysis, that is pyruvic acid, it undergoes phosphoroclastic cleavage, results are producing these that is protons and electrons, which helps in the reduction of nitrogen. And now coming to that of the enzyme dinitrogenase, and this enzyme is very particular. We can say that this enzyme is specifically seen in, or characteristically it is observed in case of the prokaryotes, or rather the microorganisms. Okay, so this dinitrogenase enzyme, which is significantly seen in case of prokaryotes, and we all know that enzymes are rich in proteins, and this dinitrogenase enzyme is rich in iron and molybdenum proteins, which is of a, we can say it is of an dimer as well as a tetramer proteins. It is rich of dinitrogenase enzyme. And this dinitrogenase enzyme, which is specially seen in case of prokaryotes, it usually, and it has a speciality. I am telling that it is present, significantly seen in case of prokaryotes, and it has one disadvantage also I can speak of, that it gets denatured in the presence of oxygen. You all know that as the enzymes are proteinaceous in nature, okay, and this enzyme dinitrogenase gets denatured in the presence of oxygen. So prokaryotes has a special, or you can say that in all the prokaryotes they have a special mechanism in order to protect, or rather in order to prevent dinitrogenase enzyme from getting denatured in presence of oxygen. So when we speak, we are not speaking about all the prokaryotes, but here we are speaking about the, what you can say, the symbiotic microorganisms. The symbiotic microorganisms like rhizobium. This is an example of symbiotic microorganisms. Symbiotic fixers, we can say, symbiotic nitrogen fixers. And example of cyanobacterian fixers. Okay, we can go with that of nostoc and anabina. Okay, when coming to that of free living nitrogen fixers, these are all nothing but the examples of nitrogen fixing microorganisms. That's what we can call them. We can simply call them as nitrogen fixers. So symbiotic nitrogen fixer, example rhizobium, cyanobacteria, that is nothing but what you call them as blue-green algae. You call them as blue-green algae in common. Okay, so nostoc and anabina are the examples of these cyanobacterians. Now coming to that of the free living. Okay, so coming to that of free living, example we can go with that of Beijeringia. Example, one of the example is Beijeringia. Okay, so in case of the symbiotic microorganisms, this enzyme dinitrogenase And symbiotic means we all know that it is present in the nodules, root nodules of Fabaceae members. Okay, here what happens here is that the nitrogenase enzyme, which is protected from that of the getting denatured from that of oxygen. Okay, here it has these symbiotic microorganisms or the leguminose members. Leguminose members, what do they have? Leguminose members have leg hemoglobin. Leg hemoglobin is nothing but a pink color pigment which is present in case of the nodule, in case of the root nodule of leguminase members. That is a specification which we can see in case of the symbiotic microorganisms. Okay, so as symbiotic association is between the rhizobium bacterium with that of the leguminase members, that is with that of the root nodules, we can say, wherein the root nodules have a pink color pigment called as leg hemoglobin, wherein this leg hemoglobin prevents the dinitrogenase enzyme from getting denatured from that of oxygen. So that's what about the specialization or the specification which we observe over here in case of symbiotic bacteria. So that's how we can say that the enzyme dinitrogenase is protected, or rather it is being prevented from getting denatured from that of oxygen by leg hemoglobin pigment. How does it act? There is a question over here. Here, leg hemoglobin pigment, what does it do? It slowly sends this oxygen levels at a moderate levels of oxygen to that of the bacteroids. Sent to that of bacteroids. So, as we all know that bacteroids are nothing but the microorganisms. Okay, and these bacteroids, as they require oxygen for the matter of respiration. So, dinitrogenase enzyme, which is prevented from getting denatured from that of oxygen, here leg hemoglobin helps in transporting the moderate levels of oxygen to bacteroids for the process of respiration over there. So, that's what actually takes place, and that's how we can say that dinitrogenase enzyme is prevented from getting denatured from that of oxygen. That's what mainly takes place here. So that is what about the second question which I have explained to you, the balanced equation of nitrogen fixation. Actually, to say this is the balanced equation of biological nitrogen fixation. Dinitrogen plus 8 protons plus 8 electrons plus 16 ATP in the presence of an enzyme dinitrogenase results in the formation of two molecules of ammonia plus H2 plus 16 ADP plus PI, inorganic phosphate. So here we have finished discussing about the second question. Okay, now we'll discuss the third VSAQ. Third VSAQ is who proposed lock and key hypothesis and induced fit theory. Okay, so who proposed lock and key hypothesis and induced fit theory? So who proposed, in the sense, obviously we have to go with the names of the scientists. So lock and key hypothesis was proposed by the scientist M.L. Fisher in the year 1988. Okay, so lock and key hypothesis was first proposed by M.L. Fisher in the year 1988, whereas induced fit theory was proposed by Daniel E. Koshland. Daniel E. Koshland in the year 1973. Lock and key hypothesis we are discussing, and we must also know that lock and key hypothesis, which was proposed by M.L. Fisher, this hypothesis has been later elaborated by two more scientists. That is, the names of the two more scientists are Paul Fields and W. Woods. These are the two scientists who have elaborated lock and key hypothesis. That's the first hypothesis in our question. Okay. So lock and key hypothesis, which was first proposed by M.L. Fisher in the year 1988. Later this hypothesis have been elaborated by Paul Fields and W. Woods. Okay. And what does this hypothesis elevate rather, or what does it explain? It explains that all the enzymes have catalytic site. All the enzymes have catalytic site, or it can also be called as active sites. Okay, so all the enzymes have catalytic site or active sites, wherein the what you can say, the substrate molecule is going to bind to this catalytic site or active site, and further the change or the reaction which takes place. And induced fit theory also explains the same, which was proposed by Daniel E. Koshland in the year 1973. So this how we have finished. Now we'll discuss next question, that is fifth question, fifth VSAQ. What is point mutation? What is point mutation, and give an example of point mutation. Okay, so here coming to that of what is point mutation. Now example of point mutation. Now point mutation, wherein mutation, first we must be first we must be aware of the word mutation. Mutation can be defined as sudden inheritable change. How does this mutation is caused? And here we are dealing with that of point mutation. And point mutation is the change or the mutation which is caused or which occurs. Okay, so point mutations are the change which occurs due to the change or alteration in, due to change in what? In a single base pair. So due to the change in a single base pair, you can say. So point mutation can be defined as change in the, or change occurs mainly due to the alteration or rather change in the single base pair of where? Where does the base pair present? The base pair is present where? In none other than that of the DNA, okay, which is the genetic material. So here mutations can be defined as a sudden inheritable change, and these mutations can be of what you can say artificial mutations or natural mutations, which can be carried out by a number of different what you can say is that the different agents, or you can say that can be of the X-rays or UV rays that can bring about mutations. These are the abiotic factors, what I have said now, that is X-rays. These are the abiotic factors which are capable to bring about mutations. Coming to that of the point mutations are the mutations or the change occurs due to the alteration or change in the single base pair of DNA. What is the example of mutation? Example of mutation over here is sickle cell anemia is the example. Sickle cell anemia is the example of mutations, point mutations. Okay, sickle cell anemia. What is the sickle cell anemia? Usually we know that RBC cells, the shape of the RBC cells, but here in these or the persons who are affected with this disease, in them the RBC will be in the form of sickle. The form of sickle, you can say. Exactly, this is the shape, you can say. Okay, so point mutation is a change, what you are telling, which occurs due to the change or alteration in the single base pair of the DNA molecule, single base pair of the DNA. And sickle cell anemia is the example. In case of sickle cell anemia, what happens? How does it occur? How? As we are telling that sickle cell anemia is the example of point mutation, how this point mutation have been occurred, which results in the formation of the sickle cell anemia. What does it form? In this case, also there is a change in the single base pair. In this case also the same, because the basic definition of the point mutation is nothing but alteration or change in the single base pair of the DNA molecule. So in case of the sickle cell anemia, there is a change in the single base pair of the 6th codon. Change in the single base pair of the 6th codon. That means here in the 6th codon there is a change in the single base pair, which codes for that of beta globin chain, wherein in case of the beta globin chain, what changes in case of the beta globin chain? In case of the beta globin chain, wherein changes from okay, so here in case of sickle cell anemia, a single base pair of the 6th codon of the beta globin chain, where the codon it is of GAG, changes to GUG, GAG to GUG. Here we must know that GAG is the codon which codes for the amino acid, that is glutamine, okay, glutamine, and whereas GUG it codes for valine. Okay, so sickle cell anemia occurs due to the change in the single base pair of the sixth codon of beta globin chain. Beta globin chain, where from the codon has been altered. That means here we can say that is the single base pair. The single base pair, that is adenine over here, has been changed to U, that is uracil. Okay, so that means automatically the codon is going to, as the code is a triplet code, and the codon which is going to code normally GAG, which is going to code for glutamate. Now if the single base pair, that is A adenine, in the place of this A which is replaced by U, now GAG becomes GUG. That means obviously GAG normally which is coding for glutamate, now it codes for valine. That leads to the sickle cell anemia. So that's what about we can say that. Okay, so point mutation is nothing but it is a change or alteration in the single base pair. This single base pair can lead to the number of differences as the codon which is, or the triplet code is going to change, and the codon which it is going to code for, it is going to code for a different amino acid. The example is sickle cell anemia, wherein the single base pair is altered over here of the sixth codon, that is from GAG to GUG. GAG which is going to which codes for the glutamate, and GUG coding for valine. So therein we have finished the fifth question.